Good morning from Toronto. My name is John Kyo, and welcome to this Future of Food mini-series for 2021. And in this series, I'm going to talk specifically around food provenance. And I'm going to talk to scientists. And right now, I have the great pleasure to introduce Professor Robert Hanner from Guelph University. Good morning, Robert.
Good morning, John. And let me say the pleasure is all mine to be here. Thank you for the opportunity.
Oh, it's great to see you and to have you. You've got a great background in this, but don't let me talk about your background. Can you, in the first minute or so, talk about who you are and a little bit about the work you do at Guelph University?
Certainly. I'm a professor in the Department of Integrative Biology. My research program is focused broadly around molecular biodiversity. So we've spent more than a decade developing a tool for species identification, commonly referred to as DNA barcoding, here at the University of Guelph. And as we've started to build this reference sequence library, it's allowed us to explore some pretty novel applications with identifying food and ingredients through DNA sequencing technology.
Wonderful. So my first question then just builds on that. For the average person listening who's non-scientific, can you explain what food provenance is and what are the main technologies or techniques that are used in the laboratory to determine food provenance?
Sure. It will, of course, be biased from my own perspective, but provenance really is focused around origin or source. It requires some documentation to establish provenance. And I would argue before we even get to provenance of where something is from is, a key part of that is what is it? Is it even what we think it is before it starts to move around? There are a variety of techniques used in food authentication, spanning simple visual analysis through to various kinds of chemical and biochemical analyses. With respect to provenance, this is usually a chain of custody documentation, but I would argue that without some verification testing to support that chain of custody documentation, that we're really only tracking the movement of packages without necessarily knowing what's in them. So I see authentication and provenance as two sides of the same coin.
Gotcha. I think that's very important. So firstly, you have technology to verify that the product is what it says it is. And I guess that's done with various technologies, specific technologies, which will look at the genetics of the product, its DNA fingerprint. And then you have another set of technologies that would look at the trace elements or the composition of the soil that they were grown in.
Sure, the terroir. Yeah, and as you mentioned, there are many different techniques. The work in my lab is focused almost exclusively around DNA-based methods.
Gotcha. So when you look at the value then of provenance for the food industry, what do you see as the main value for the food industry? And can you pick a product and then talk about the actual technology that would be used to prove its provenance?
Sure. I think the number one value proposition for industry is to protect brand reputation. But there are other relevant issues here. Of course, there are, I would say, three parts around food authenticity and safety that I think about. There are, of course, the economic aspects. Depending on where in the supply chain you are, if we see misrepresentation of products, you may not be getting what you're paying for, whether you're a processor, a retailer, or even the end consumer. We've also seen some environmental outcomes that relate back to provenance, where sometimes we see that we're over harvesting some wild species, and if there isn't a good chain of provenance, we may not know which population they come from. So one population may be sustainably managed, another not. And over-exploiting can, of course, lead to resource depletion, possibly ultimately even extinction. So in addition to economic and environmental concerns, we also have to think, perhaps most importantly for us as individuals, are about what are the human health implications around provenance. If it's not carefully tracked and the products are not properly handled, we can see foodborne pathogens making their way into the supply chain. But coming back to authenticity, if we're not careful about what we're actually putting into a product, there can be chemical contaminants, biochemical toxins of a given species. So there are many different aspects of human health that depend on proper authentication and provenance documentation.
Gotcha. Now, if I go back to a number of years, I think when we met first, maybe a dozen years ago, you were one of the leaders in this breakthrough around fish fraud. Now, I don't want to talk about fish fraud in particular, but around the speciation. Can you talk about your work in fish, around provenance of fish? I mean, you mentioned one of those with regard to environmental and sustainability aspects of fish populations. But can you talk about the work that you did in the area of DNA barcoding and the value of that, and the challenges that brings?
Absolutely. First and foremost, I would say species exist because they evolve. And the whole barcode of life movement was predicated around finding a particular gene region that seem to be relatively invariant within species, but variable between them, such that we could genetically characterize expert identified reference material and then use that as a lookup table by sequencing this marker gene of an unknown, comparing it to our library, we could infer an identification. That's of course important in things like seafood, where once a product has been delayed, possibly breaded and packaged, the classical morphological features that we would use to identify a species have been removed. So that sets the stage for potential misidentification or even deliberate misrepresentation of products, because we see that some species have a higher economic value than others. And this differential in price sets up a potential motivation for fraud. So my original role in the Barcode of Life Network was to work with fisheries biologists around the world to develop this DNA barcode reference sequence library to help us identify fishes. And we use this in a number of ways academically, from identifying eggs and larvae to identifying stomach contents to know who's eating whom in these aquatic systems. But interestingly, we started to wonder whether or not our reference database was mature enough that we could begin to identify fillets commonly encountered in the marketplace. So our initial foray was to look at samples coming from New York City and the greater Toronto metropolitan area. And what we began to see was that, yes, our database was mature enough that we could identify all of these fillets we were encountering in commerce to the species level. But what was rather surprising to us in that initial study was that about a quarter of the fillets we tested were not the species they were purported to be. And in nearly every case, it was a species of a lower economic value being substituted for one of a higher economic value, suggesting economically motivated adulteration or fraud. We've subsequently repeated these studies in North America and elsewhere and found that this issue of seafood fraud is endemic across the entire supply chain globally.
Gotcha. Now, I have a question for you because I don't know the answer to this. We know that there's all these fraud issues and various rationales behind it, economic motivated adulteration. And I believe as you go closer to the consumer in fish, the fraud increases. But and so I'm going to ask you a question if that's true based on research. And secondly, Is it still possible when the fish is cooked, if it's presented to you in a restaurant, can you still do genetics and identify that species when the fish is cooked? Or if it makes somebody sick from the contents of their stomach as part of an epidemiological analysis?
Great questions. So we do see that a certain amount of processing will degrade DNA. This just happens over time. But because we're focused on a very short fragment of DNA, it tends to remain intact through various processing steps. So we can barcode seafood, whether it's been frozen, cooked, baked, broiled, fried. It doesn't seem to matter. The only issue we've encountered is the combination of high temperature and pressure will break that DNA down such that a classic DNA barcoding approach of sequencing the marker gene often doesn't work. In other words, the DNA is in too small of fragments to use a DNA sequencing approach, but we do have other methods of developing short molecular probes that exploit species-specific patterns of variation in the targets we're interested in, such that we can still authenticate even canned products. So the technique is quite resilient to processing.
Wonderful. Now, I'm often told that I have a long and good memory, and I have a memory of one of our conversations maybe 10, 12 years ago. And correct me if I'm wrong on this, but you were asked to investigate a food safety outbreak or an issue with a food product in Canada. It was, I think, a salad, and there was a piece of a mouse identified in a chicken salad, and you were able to track that mouse back to its provenance, which is back to, I think, Thailand. Is that the type of science that we can expect in the laboratory? Is my memory good?
It is. That work was done by some of my colleagues at our Center for Biodiversity Genomics here on the University of Guelph campus, where a food processor in Toronto was importing chicken from Vietnam, and one of the people in the processing plant, as they were preparing these prepackaged frozen meals, noted these fragmentary remains, as you mentioned, the mouse head. And so they, of course, stopped the production line. And the key question for this plant was, is this a Vietnamese mouse? Hence a problem coming from our chicken supplier that goes into these prepackaged meals. Or is it a North American mouse such that it's a problem in our plant? So to try and determine how best to react to this issue, this kind of provenance information about the contaminant, in this case, was one that we sought to investigate with genetic techniques. And while DNA barcoding is typically focused with species ID, we do often see patterns of phylogeographic variation. In other words, populations that aren't interbreeding in different parts of the world will accumulate small mutations that can often allow us to separate these lineages within a species. And in this case, we were in fact able to show that it appeared to be a mouse of Asian origin and not North American.
Wonderful. So I think I got my memory of the story was good, but just Thailand versus Vietnam and living there. Wonderful. So Bob, I know we have a time constraint this morning, but with regard to the database, the reference database, do you think that's the way to go worldwide? That let's say all the Canadian products that we can sequence to prove that a product is from Canada or a particular region in Canada. Is that the way to go? And should that be shared with competent authorities and buyers around the world so they can use that information to verify that a claim that a product is from Canada or from Saskatchewan or from Alberta, that it's true?
Yeah, this is becoming a very powerful technique. So our DNA barcoding project is really an academic exercise to discover and characterize all species around the planet. So this hasn't been restricted just to Canada. The tools that we've been developing using classical Sanger sequencing of this DNA barcode marker have been utilized by the U.S. Food and Drug Administration and the Canadian Food Inspection Agency, FARA in the UK, and others to authenticate seafood. But as our libraries are developing for plants and other products, it becomes a powerful tool to authenticate an ingredient. But the challenge with classical Sanger sequencing is that it works on something like a discrete fillet, but it doesn't work on mixtures. We can overcome this by employing high throughput sequencing technology, where we can simultaneously barcode everything in a mixture to begin to identify ingredients. Now, this was some of the work we did with Canadian Food Inspection Agency looking at ground meat, where they sourced samples that were purported to be of a single species, and we used high throughput sequencing analysis and quantitative PCR methods to show that in about 20% of the products that they sourced across Canada, there were traces or even cases of complete substitution in some of these products. So we were seeing significant amounts of pork and ground beef and vice versa, which is of course of concern to our kosher and halal consumers. We were also seeing cases of so-called ground turkey being wholly adulterated with ground chicken, which is of course a cheaper commodity. So this is where we've started to work on being able to identify mixtures and pick up potential contaminants, as well as these kinds of tools can be used to identify foodborne pathogens. But one limitation we face is this issue of provenance. In other words, can I tell a Mexican bell pepper from a Southern Ontario bell pepper? If they're both using the same seed stock, they're going to have identical barcodes. So we can't actually establish geographic origin in many of these commodities where seeds are being moved around the world. Mouse was a different story. We're talking about essentially natural lineages that are reproductively isolated. For many of our crop commodities, they're being, the same seed is being used all over the world. So we can't directly query provenance by sequencing the barcode of that product. But what we can do, and what is starting to emerge as a powerful tool, is to actually swab the surface of these products, use high throughput sequencing to characterize the bacteria on the surface of a product. And what we find is that there are some bacteria that, and could also even be pollen grains or fungal spores, that will be endemic to a certain region of the world. So in these cases, there may be some bacteria that are broadly distributed, but there will be others that are characteristic to certain regions, such that we can also use these DNA-based tools to start to investigate provenance by looking at what we're calling the food microbiome. And that's been quite interesting to see. In Europe, there was a recent study where they were looking at the same species in a number of different aquaculture operations across Europe, but they could actually pick up different microbial signatures from the different aquaculture operations to help support not only authentication, but provenance.
Gotcha. Very interesting, Bob. So you have the DNA sequencing of the product, you have the swabbing to look at the bacterial. Is it also possible then to do the trace element analysis, so to be able to associate it back to chemicals found in the ground, or is that also possible with using trace elements?
Yeah, this idea of biological tracers is something that I'm exploring with colleagues here at the University of Guelph to pair with DNA-based methods. The challenge in doing something like that is very similar to the challenge we faced with DNA barcoding. We need to source material from around the world to build reference chemical profiles against which to identify unknowns. And that's obviously a non-trivial undertaking.
Yeah, of course. And the point you mentioned earlier, the work you did with CFIA in looking at a particular product, and you found multiple DNAs in there, multiple species in there, and you mentioned, of course, how offensive it could be to faith-based groups if pork was included. But I believe in some cases this is actually legal. Is that an issue?
Well, that's a really good question. I mean, each jurisdiction will have its own regulations. Our interpretation is if there's a single species listed on the label, then that's what you should be getting. So when we did this kind of mixture analysis, we did consultations with both the CFIA here in Canada and industry to say, what is a meaningful level of contamination? You know, these molecular methods are very sensitive. We don't want to put people on high alert for tiny amounts of, say, trace carryover from, say, potentially grinding pork sausage one day and beef the next. And what we got in response from industry was that, well, if you have more than, say, 1% adulteration, if you've got more than 10 kilos per thousand in your grind, you clearly have a supply chain issue. That isn't trace carryover. So we arbitrarily set that threshold of what we reported as a positive adulteration at 1%. So that it would be more indicative of a supply chain problem than perhaps a trace contamination issue in the case of the ground meat. So we obviously want to work with regulators to determine what are appropriate thresholds. We do know that, for example, if we're using high throughput sequencing, plants grow in the ground. We're going to potentially pick up some fungal spores and pollens from other plants, and that's normal and to be expected. But if we start to see a large adulteration of alfalfa and, say, herbal tea, then that's just a cheap filler that's being used to bulk up the tea bags. We've seen evidence of that too. In fact, as colleagues have worked on food authentication across the spectrum, we see this in nearly every commodity. It isn't limited to ground meat or seafood. It's of course well known in honey and dairy that there can be adulterations. Similarly, olive oil. So it's been fascinating to me to see how many different commodities we're finding significant amounts of adulteration. And I think whenever we see supply chain squeezed, as we're seeing now under the current pandemic with labor shortages and things, it only exacerbates that tendency for economically motivated adulteration, either because of supply chain issues or financial hardship. So I think these are increasingly important. And, you know, there's been some question, at least around this seafood work we've done here in Canada, as to whether or not this is purely a problem with international suppliers or if it could be happening here in Canada too. And all of our prior work on seafood fraud had been at the retail level, because as a consumer, that was all I could source. But working with the CFIA, they actually source fillets at import, at wholesale and processing plants and at retail, and sent them to us for testing. And what we found was that about 20% of the things coming into Canada were mislabeled. That jumped up to nearly 30% when we began to look at wholesale and processing, and then nearly 40% by the time we got out to retail. So what we were seeing is that there was this kind of mislabeling occurring both farther back in the supply chain as well as here in Canada. And it seems to amplify at each point in the supply chain, suggesting that if we are really concerned about authentication and provenance, that we do need some testing at multiple points in the supply chain.
Yeah, I think that surveillance is a very important point, and thanks for sharing that, Bob. My last question for you, because I know I'm sensitive, you have to jump on another call, around gathering the evidence that would stand up in court. Is that something that you see maturing as an area where the courts would accept, or CFIA, if they do bring a case to court, that the evidence from laboratories is now being accepted?
Yes, as these techniques have shifted out of academic labs and into ISO accredited laboratories that routinely maintain chain of custody documentation, this kind of evidence is essentially no different than any other, as long as it's done in a properly accredited facility.
Wonderful. Folks, there you go. There you have it, all the way from Guelph, Ontario, my good friend, Professor Robert Hanner. Robert, thank you so much for sharing your knowledge and wisdom with us today, and look forward to working with you in 2021.
My pleasure, and thank you as well. Always happy to collaborate. So thanks again, John. It's nice to see you.
Thank you, Bob.